Do fish produce too many offspring that the environment can support? Sure, each fish will lay thousands of eggs, and most of them will die out, and only a few will actually reach adulthood. So there will be competition for resources. Second one, there is variation in the population. Is there variation in the population? Yes, we have two different versions of this enzyme. So we have some ancestral enzyme form, and then a mutation happened, a new enzyme form appeared. So there is variation. Is that variation heritable? Sure, because that's a protein, and changing amino acid sequences of the protein mean there was a change in the DNA coding for that, so it's absolutely heritable. Differential survival and reproduction. Will there be differential survival of these fish? Yes, why? Yes, sir.
The fish that LDHA can survive better in the south because of that environment, while the other ones can't survive. So in the north, the environment sells off the eggs and fish.
All right, so depending on where you're going and how what the temperature in the local age is, one enzyme type might be better for you. What do these enzymes do? And you have no idea because it just says M4LDH. But it is a muscle enzyme. So if it's working properly, then your muscles are functioning properly. And how would that help you surviving better? If you're a fish, swimming, eating, escaping predators, all of those rely on your muscles working properly. So if your muscles are working properly, then you have a better chance of survival. And also, if you have functional muscles, then you have a better chance of mating. Because you have to compete for mates. You have to, if you're a male fish, you have to provide a nest for the eggs. The males usually make the nest, the females lay the eggs, and then the males have to defend the nest against other males. So the males have to do a lot of work with the fish. So if your muscles are not working properly, then you won't reproduce as well either. So there will be differential survival and reproduction depending on how functional your muscles are. And that means that whichever version of the enzyme works better for you will be passed on more often to the next generation, the next population. So when you look at the southern populations, you get more of the LDHA type, and in some places only that one, so all of the D has been eliminated. And as you move further and further north, you get more of a mixture, and then you get proportionally more and more of the D type. So far, so good? Great. So when we talk about enzymes, we sometimes make graphs like this, and in lab, in a few weeks, we will do that, although not as complicated as this one. When you take biochemistry, you will do a lot more than this. And for now, all we want you to know is that when you look at a graph like this, you're looking at the effect of substrate concentration on the rate of activity of the enzyme. So if there are no substrates, there will be no reaction. So the rate is 0. So that's this one. As you add more and more substrates in the presence of a given amount of enzyme, the reaction rate goes up because now more and more substrates can be used and more and more products will be produced. Until you begin to plateau, because now all of the enzymes are working as fast as they can and their active sites are occupied all the time, and adding more substrate doesn't help anymore because all of the enzymes are occupied. That's like if all of you were holding a ball and then I start throwing more balls at you, but you can catch it more because you all have your ball. But if only half of you have balls, so if you're down here, I did not come out right. Half of you had tennis balls, and I started throwing more tennis balls at you, then you could catch more tennis balls. So the rate could go up. But if you all were holding tennis balls in both of your hands, you couldn't catch any more. So the saturation point means that all enzyme active sites are occupied all the time, and adding more substrate doesn't help anymore. Below the saturation point, as you add more substrates, more enzymes can use those substrates, and therefore the rate of the reaction will increase. Now, the Vmax is the saturation point, and then the Km is the concentration of substrate at which the rate of reaction is half of the maximum. And these two terms are defined in your textbook, so if you don't write it down here, that's okay, because you have it in your textbook. So just remember, this is the maximal rate for a given amount of enzyme, and the Km is the concentration of substrate at which the rate is half maximum. And if you know these two terms, you can figure out what the shape of this curve would be for your enzyme. Now, we will not ask you to calculate Km or Vmax in the class. However, when you do the enzyme lab, you will generate graphs and you will see the saturation point. What we do want you to be able to do is distinguish what will happen to this curve if there is an inhibitor. So we're going to talk about different types of inhibitors. But before I go forward, any questions about the general shape of this curve? So the two main types of inhibitors that we talk about are competitive inhibitors and non-competitive inhibitors. Competitive inhibitors bind to the active site. So they're competing for the active site with the substrate. So sometimes the substrate binds to the active site, sometimes the competitive inhibitor binds to the active site. The non-competitive inhibitors typically bind somewhere else. They don't bind to the active site. And they cause a change in the shape of the enzyme. So the active site no longer looks correctly, and therefore it cannot bind to the substrate. So the change in the shape of the enzyme is the reason the non-competitive inhibitor works, as opposed to blocking the active site because it's occupied, which is what the competitive inhibitor does. And they have different consequences for what the curve will look like when you use competitive as opposed to non-competitive inhibitors. This diagram is showing you what we mean when we say competitive binds here, non-competitive binds there. So here's your active site. Here's the substrate. Normally the substrate binds there for reaction. But if you have a competitive inhibitor, that can bind to the active site and will block the active site. So sometimes the competitive inhibitor is binding, then dissociating. Sometimes the substrate is binding and the reaction goes forward. So what happens as you increase the concentration of the competitive inhibitor? So if you increase the competitive inhibitor more and more, what will happen to the rate of the reaction? It should go further and further down. So for the same amount of substrate. What if you have the same amount of inhibitor and you keep increasing the substrate concentration? Yes, sir.
The curve should remain more or less the same, like at least maintain.
As what?
As it would have been if the substrate level was what it was previously before introducing the inhibitor and the extra substrate. So if you consider how many substrates you have before introducing the competitive inhibitor and the extra substrates, when you introduce the competitive inhibitors and all the extra substrates, then the rate of reaction should maintain the same curve as before, because you're increasing the competitive inhibitors and you're also increasing the chances that a substrate would bind to the site.
So if this is your normal curve, no inhibitor
This color.
of reaction,
Caramel.
and this is the substrate concentration. No inhibitor. This is the normal curve. That's your Vmax. Let's say you now add a known amount of competitive inhibitor. What would that do to this curve? What up or down? why would it shift all of it down? Talking about a competitive inhibitor that is going to compete for the active site. So we are talking about so you have a known amount of competitive inhibitor, and you keep increasing the substrate. As you keep increasing the substrate, what are the chances that the substrate will bind as opposed to the competitive inhibitor? Keep increasing the substrate concentration. Who has a better chance of binding? The substrate. If you had low substrate concentration, then the competitive inhibitor would have a good chance of binding. But if you keep increasing the substrate concentration, the chance that the inhibitor binds goes down, the chance that the substrate binds goes up. Yes, ma'am?
How long does the competitive inhibitor bind for?
So there are different types of inhibitors. Some inhibitors bind covalently and stay forever. But generally, when we talk about competitive inhibitors, they bind, dissociate, just like substrates can bind and dissociate. So it's not a permanent binding. It's not a covalent bond, usually. Because if it's a covalent bond, it's no longer competitive. Yes, ma'am. Very good question. Affinity always comes up. So for now, for our case, we're assuming equal affinity. The only thing that will matter is the concentration, or what we call the effective concentration. Yes?
Does the inhibitor affect the activity of the enzyme?
The inhibitor just binds to the substrate. Doesn't change the shape of the enzyme in the case of the competitive inhibitor. It just binds here, releases, binds, releases. Just blocks the active site when it's bound. So as you increase the substrate concentration, what will happen to the rate of reaction? So you have to think about that in terms of that curve. The other option is that you use a non-competitive inhibitor. The non-competitive inhibitor, which is referred to, hold on, if it's referred to allosteric, means that it's binding somewhere other than the active site. So this is allosteric, it's binding somewhere other than the active site, and it's causing the shape of the active site to change. So the substrate can no longer bind there. So if the substrate cannot bind, then it doesn't matter if you increase the concentration of the substrate. If you've changed the shape of the enzyme, it doesn't matter how much substrate you have. No, because none of them will bind. So with the non-competitive allosteric inhibitor, the concentration of substrate is usually irrelevant because the enzyme has changed shape. Non-competitive, the competitive inhibitor, the enzyme has the right shape. So if there is substrate, it can bind. You just have to make sure that there is enough substrate to compete well with the competitive inhibitor.
So would it change the shape of the curve from like the extreme block curve you have now to a flatter slope?
So we are going to take a look at that. But let me just give you a few examples first. So many poisons happen to be enzyme inhibitors. So a lot of pesticides, you're going to do a lab using a pesticide as an enzyme inhibitor. And you will be wearing goggles and gloves and fully covered from head to toe basically when you do this. Even though we use very, very, very small amounts in very dilute solutions. And the only way for you to be effective would be to drink the whole bottle, which of course you will not do. Because if you do that, you will have plenty of other problems than just the insecticide you're drinking. But you will take a look at one example of a pesticide as an inhibitor. But a lot of other poisons that might poison you also act that way. So sarin inert gas, which has been unfortunately in the news in the past couple of years for one of the wars around the world, is a specific inhibitor of an enzyme. So acetylcholinesterase, which happens to be the enzyme you're going to study with the insecticide, sarin inert gas can bind to an R group of one of the amino acids in the active site. So this is a case where it actually binds covalently. and it will inhibit that enzyme.